Hi, everybody. Good morning. Thanks for coming. I found it cruising around the campus, but I am clearly a clinician, not here very much. My name is Veronika Bachanova. I am an associate professor of a division of hematology and oncologic transplantation. I am a translational and clinical researcher. So most stuff I'm going to talk to you about is of course clinical, but I will talk to you a little bit about the path towards the clinical trials which we are doing. And so we'll talk about enhancing immunotherapies for hematologic malignancies. My specific focus is lymphoid malignancies, lymphoma, and I will talk to you about a path through some solo therapies all the way to more immune engagers, bike and trikes treatments. So my target disease is non-Hodgkin lymphoma. For those of you who are not in a clinic, this is a cancer of lymph nodes, of stems originates from the lymphocytes. It is the most common hematologic malignancy. It's sixth most common cancer. And it really is very treatable. However, we still have a long way to cure this cancer. As I said, this is the second, the most common hematological cancer. And it's more common in Minnesota than other states. It's a very complex disease. It has close to 50 different subsets. The differences are all the way from genetics, epigenetics, to specific histological subsets. And mostly clinically, there are two large groups of indolent and aggressive lymphomas. And I just want to point out that what we do in immunotherapy is really quite crude right at this time, affecting sort of trying to target the lymphoma as a group. But as we learn more about subsetting, I think we are going to learn about how different subsets perhaps are more or less responsive to immunotherapies. As I said, for not a clear reason, the age-adjusted death rates and incidence rates of non-Hodgkin lymphoma are higher in Minnesota than in some other states. There are about 1,400 new cases of non-Hodgkin lymphoma in Minnesota each year, and about half of these patients will succumb to their disease. Just to really very clearly show you on this, what's called a Kaplan-Meier curve. These are the survival curves of the patients, in the populations of the patients with lymphoma in three main subsets: large cell lymphoma, follicular lymphoma, and mantle cell lymphoma. And the point of this curve is really to demonstrate where we are with the current therapies. The chemotherapy sort of reached the ceiling of the efficacy. No matter what we add or how well we combine cytotoxic agents, we are really, as you see with this comparison arms of different treatments, different new additions, we are not making more progress. And so there is a need for really clearly developing new therapy, which has been happening over the last decade. So about 60% of large cell lymphoma will be cured. Follicular lymphoma and mantle cell lymphoma are not really curable. The survival is about 60% at 10 years, but eventually almost all of these patients will have a relapse disease. Mantle cell lymphoma is 50%, and this is a disease which is even more aggressive. So how can we enhance the survival? As I said, the chemotherapy, we probably will not get much out of the chemotherapy or cytotoxic therapy. So with the invention of the monoclonal antibody, rituximab, the field of immunotherapy started about 18 years ago, when the first monoclonal antibody, which was rationally designed to target CD20 on the surface of lymphoma cells, have proved to be effective on its own, and it's now a mainstay of all combinations for B-cell lymphoma, has not only deepened the remission, improved the remission rates, but really impacted the survival. And there's a number of antibody, this is a naked antibody, but antibody conjugates, combining antibody with drugs, toxin, radioisotypes, and other ways how to improve their efficacy. There are checkpoint inhibitors, which is one big group, and many of you are working in this field, which I will not address today. But what we mostly will talk about is natural killer cell and T-cell immunotherapies and immune engagers, which link tumor with immune effectors. And there are really ways to try to simplify the cell therapy, but to use small molecules which can link the immune system with the cancer. So most of my talk will be about natural killer cells, and these are the innate lymphocytes circulate in the blood. About 10% of lymphocytes are NK cells. They are they don't need priming to be active and to recognize the cancer, and they are very potent cellular killers. So we'll talk about NK cells and understanding how they work. We will review some clinical experience with the NK cell therapy in lymphoma, and then I will talk to you a little bit about the CAR-16, which is one of the novel ways how we can use the chimeric antigen receptor reprogramming. So the NK cells, as I said, are innate lymphocytes. They don't have a TCR, they don't need prior antigen recognition, and they kill with 3 different stimuli. They can be activated with cytokines, mostly IL-15 and IL-2, which they have receptors for, as many of you in immunology know, they have receptors for IL-15 and IL-2, and they get stimulated, activated, release cytokines, and can kill. They very importantly mediate antibody-dependent cell cytotoxicity. So they have a receptor for a Fc fragment of monoclonal antibody, CD16, and this is the most potent activating signal, which leads to NK cell activity, proliferation, and killing with either interferon gamma or production of a perforin and granzyme, the cytotoxic granules, from cytotoxic granules. The third way how the NK cell kills are through the natural cytotoxicity, and this is by them expressing a number of activating and inhibitory ligands, the most specifically killer immunoglobulin-like receptors. These ligands are either inhibiting NK cells if they see the ligand, which is HLA class 1, or they are activating receptors which can activate NK cells with the neo antigens which are expressed either on virally infected cells or tumor cells. So these are 3 different ways how the NK cells get activated. In an autologous setting, so your own NK cells are under tonic inhibition of your own class 1 HLA. So they are really not active and they don't really affect or impact our normal cells. However, in a viral infection or or tumor transformation, there's either a loss of class 1 HLA or the expression of the neoantigens, which then activate the autologous NK cells. But this is often not really effective, and the HLA loss can be only partial, and the autologous NK cells remain dysfunctional. The allogenic NK cells, so using natural killer cells from the donor, are better killers, and that is because they are not inhibited by your autologous class 1 HLA, and also there's a high proportion of the NK cells which really are able to induce this cytotoxic response. So we have explored this concept in a bone marrow transplant, of course, but we really have been interested in this in an adoptive setting. This is a slide just demonstrating to you how are the NK cells dysfunctional in patients with lymphoma. We've looked at several patients and comparing healthy controls with the newly diagnosed patients and patients with refractory lymphoma, and this is looking at the functional assays of the NK cells. As you see, the granulation, the degranulation assay, which reflects the killing of the NK cells, is significantly impaired in patients with lymphoma, as well as the interferon production and CD16 expression, which would mediate ADCC. And there's also difference in expression of immunosuppressive molecules of the NK cells, such as TIGIT and TIM3. So the NK cells are lower in numbers, but also they are dysfunctional and hyperfunctional in cancer patients, often unable to control the lymphoma. So we have developed this concept of adoptive natural killer cell transfer to cancer patients under leadership of Jeff Miller. And this is a platform how we do it. We have a patient and a donor who is haploidentical, so half matched in HLA class 1 and 2. The patients are receiving a low-dose chemotherapy. This is to deplete the autologous lymphocytes, to suppress immune system so they will be able to accept those adaptively transferred lymphocytes. The donor is undergoing peripheral blood leukapheresis. Thus, that product is depleted from the T cells, CD3 depleted, and sometimes CD19 depleted, incubated with the cytokines, most often IL-2 or IL-15. And after 24-hour incubation, these NK cells get a little more activated, and they are infused to the patients after he received this chemotherapy. And this is the dose which we have been using. And we follow this therapy with subcutaneous injections of IL-2 to really try to ensure the persistence of the NK cells in the blood. And we've published the initial experience more than 13 years ago, and have been really exploring this platform, how to improve it, how to make this work better, and eventually to turn this into a potentially curative therapy. So in AML, this has been a proof of concept, a disease where this therapy works. This is the patient with AML. This is how the bone marrow biopsy looks at the diagnosis, which is filled with myeloblasts, immature white cells causing the leukemia. And after the chemotherapy and NK cell infusion, the marrow is emptied and is free of leukemia. And if you look under high power, you can see the natural killer cells in the bone marrow. And these are the donor-derived natural killer cells. And this therapy has been partially effective for many patients. And I've brought this concept to the lymphoma and developed several clinical trials to really evolve and develop this therapy for the lymphoma malignancies. So first trial was used allogeneic natural killer cells, and just because we have antibody and we know that NK cells induce the ADCCs, in this disease we combined with rituximab as a form of enhanced anti-tumor targeting. So as you know, the rituximab binds CD20, and this is all B cells, cancer B cells, and the physiological B cells, and the NK cells are effectors, which are mediating the killing of the B cell through this mechanism. So this is the design or schema of this clinical trial where we enroll the patients. They are given this lymphodepleting chemotherapy. We give them rituximab for 4 different injections for weekly. The donor cells are prepared here and they're infused in a single infusion, and then we give the patients IL-2. So we have used this for patients with CLL, which is another type of a lymphoid malignancy, and a non-Hodgkin lymphoma. And these patients are really those who are not longer responding to chemotherapy, those who fail their standard approach treatments, and they are having a progressive disease on chemotherapy. These are what we see in a product. And when we have a product apheresed, it contains very few NK cells, 12%. That's just about what we have in the peripheral blood. With the CD3 depletion, we have about 39% of NK cells in the product. With the activation, the best we can achieve is 40 to 50% frequency of NK cells in the product. So there are lots of other cells. And in fact, we don't exactly have those. I mean, we know there are monocytes. There are some V cells. And there are no T cells. As you see, the T cell content is less than a couple of, hundreds of percent. But this is the cell dose we were able to achieve. And these NK cells are very cytotoxic, and this is pre-activation. So the IL-2 indeed improve their killing ability. So we are giving the patients the best product we can achieve under these circumstances. What we can see is that the NK cells persist. They indeed, after infusion, stay in a patient's blood. This is looking at NK cells, and here are the T cells. If you look at the donor HLA versus recipient HLA, as in this picture, the donor, this is the discrepant HLA, and we can detect the donor NK cells in the blood at day 7, and these are 7 individual patients, and pretty much everybody has at least few of these NK cells available. However, they don't persist for very long. And in many patients, what we see is that instead of the NK cells, in fact, at the baseline, we have very few T cells and very few regulatory T cells in patients' blood. At day 0 after chemotherapy, most of the lymphocytes are depleted. However, at day 14, after the IL-2, what you see really that expansion, the IL-2 also result to expansion of the host regulatory T cells. And indeed, these regulatory T cells are expressing FOXP3, they are CD127 positive. So these are functional T cells, they are functional assays, and they are functional, and they are in fact inhibitory for NK cells. So this is one of the barriers. The IL-2 is really causing this, and as a result of this findings, we have worked to reduce the dose of the IL-2 and also replace the IL-2 with other cytokines, and the IL-15 become available, and also IL-15 supra-agonist called ALT-803, which have been used in some of the other clinical trials, and mostly for AML. For this particular project, we have 4 objective responses in 14 patients, about 30% response rate, and these patients were very refractory. So the complete response lasting for several months, and we found some circulating donors. The good news is that it was very well tolerated. This is no toxicity with this therapy, no GVHD or cytokine release syndrome. So we had a platform which we could build on and improve. One of the correlative science results or findings are demonstrated in here. So we compared the NK cells in those patients who responded versus who didn't. And what we found that indeed in the responding patients, the NK cells are proliferating better based upon the Ki-67 staining. These patients also have a higher secretion of the endogenous IL-15 at day 0, which is at the time of NK cell infusion, and also after chemotherapy. So these patients somehow have responded better to chemotherapy and have endogenously increased levels of IL-15. And indeed, the IL-15 in the blood correlated with how the NK cell expanded. So what this tells us that it's important that the lymphodepleting chemotherapy is important, the endogenous IL-15 is important, and we need to try to find the ways to increase the IL-15 levels. This is clearly dose-dependent. We try different chemotherapies, and with a higher dose of chemotherapy, especially Cytoxan, you do have a high IL-15 levels. However, it also can lead to higher toxicity. So there is a very fine balance there. We also find that myeloid-derived suppressor cells and regulatory T cells are increased in patients who did not respond. And this is both prior to therapy and post-treatment. This is for the Tregs, and this is for the myeloid suppressor cells. And this is possibly one of the mechanisms that this is something about the immune system of these patients which really predefines whether they will respond or not. And it may not be that much with our intervention. However, we can adjust intervention if we understand the underlying mechanisms of immunosuppression. Here is, again, the negative correlation of the Treg versus the proliferation index for the natural killer cells, and the same with the myeloid-derived suppressor cells. So if we can find ways to eliminate Tregs and MDSCs selectively, there is one of the potential pathways how we can improve the efficacy of this therapy. So we are limited by short-term inpatient persistence, by limited in vivo proliferation, possibly impaired migration, and we are working on trying to look for these NK cells in the lymph nodes and the tumors of these patients, and we can find it in some patients, but not in all. And then interference and inhibition by the recipient immune system. So having these findings, we paired and collaborated with one of the companies which is developing a new way how to renew the stem cells, and this is using nicotinamide, which is the regulator of NET-related signaling pathways, and there is a very good preclinical evidence that this particular compound improves many processes of the renewal and proliferation of the hematopoietic and progenitor cells. So the nicotinamide affects the NET precursors. It affects the NET precursors. It works in this enzymatic pathway, and it is probably helping with the cellular stress, especially with those cells which have to be cultured ex vivo. And it also has impact on epigenetic regulation and histone acetylation. This company is working mostly with the cord blood-derived stem cells and expanding those in vivo for the cord blood stem cell transplantation. So there is interest in this product, and in a cooperation with Gamida, we had a number of experiments to see whether we can improve NK cell viability, function, and persistence. They developed a process where we use the same initial product, which is the leukapheresis peripheral blood from the haplomatched or mismatched relative donors. There we CD deplete these products, and we have a heterogeneous cellular product here. And then after incubation with the nicotinamide and IL-15 in human serum, there is a enrichment of the NK cells, outgrowth of the NK cells, and after about 14 to 20 days of culture, the NK cells comprise 98% of this product, and they are very functional. This is what happens in an epigenetic and genetic level. There is a major difference in expression of multiple genes. About 200 genes are differentially expressed, and these are mostly to do with the cell cycle, with the cell proliferation, with the metabolism, and also the activation of the immune response. And I really didn't didn't show you all the genes, but mostly the pathways which are affected. And this is a substantial modification which is meaningful for the function of these cells. What we found in this experiment is that there are several different changes in expression of the important receptors. This is the expression of CD62L, which is L-selectin, which is allowing the NK cells to migrate and home into the marrow and the tissues, and this receptor is substantially improved in expression. In terms of the function, if you culture the NK cells with the IL-15, they undergo exhaustion, and they have a worse production of the interferon, and they actually have also increased production of the Fs ligand. However, with the nicotinamide, this is prevented, and they are improved in their function, but also we are able to get a higher number of these cells. The third important function is the mediating of the ADCC, and with the co-culture with rituximab, the NAM-incubated NK cells are almost twice as potent to mediate Reg cell killing. So we brought this product into the clinical trial again, and we enrolled a patient with a non-Hodgkin lymphoma and also multiple myeloma with a similar preparative regimen and with expanded NK cells. And we decided to use 2 separate infusions of the NK cells, about 2 days apart, and then also combine them with either rituximab, which is for lymphoma, and elotuzumab, which is a SLAM7 monoclonal antibody, which is targeting myeloma cells. So we have now 2 diseases, non-Hodgkin lymphoma and myeloma, with a similar concept to really try to broaden our understanding whether there is also some differences in a disease and how they could respond to this therapy. So these are the 3 dose levels. This particular technology allows us to increase the number of NK cells we can use, and these are also very pure products containing 99% of NK cells. So if you look at these dose, and we are currently at dose level 2, this is the dose 10 to the 10 to the power of 8, which is about 5 to 10-fold higher what we could achieve with just regular apheresis from this first trial. So we really are making more NK cells and hoping that can result in their better persistence. So again, a similar inclusion criteria, very refractory and relapsed patients who are really at the end of their options for treatment. So these are the product characteristics. We so far treated 7 patients, some of the with the lymphoma, 5 and 2 patients with myeloma. And really just to show you that we are able to achieve pretty good doses for the total nucleated cell, which is mostly NK cells. In these 2 patients, we would not be able to achieve 3rd dose level, and we are moving towards that. And I started to worry about whether we will be able to get a 3rd dose level. These 3 patients in red here, they would have a 3rd dose level if we only look at the NK cells, but their number of T cells is slightly higher than in a previous production of the product, and now we are above the limit of the T cells we allow in a product because the potential of the GVHD. So we are working with our collaborators to try to improve the production so we actually can move to the third level. For right now, we really are we are limited. These cells are very powerful. Look at the cytotoxicity of the cells when they are of the NK cells after apheresis. They are really not very potent. They have to be activated, and after activation, this is the product at the first day, and this is the product on day 3, and both of them has about 80 to 90% cytotoxicity at the different ET effector to target ratios. The donor NK cells are detectable in the peripheral blood of the patient. This is one of the examples. This is patient number 002, and we are finding the donor NK cells in the blood. However, again, they do not persist. The NK cells are there in day 4, day 7, and they are anywhere from mostly at day 4, and then by day 14, we are not really able to detect these NK cells. However, we only look into one compartment, and that's blood. And we know these particular NK cells are very high expression of the homing and trafficking receptors. So it's possible that they are in a lymph node, and that's why really they need to be, because this disease doesn't have a marrow or blood involvement. So our next step, and what we are doing, we have a biopsy of the patient and looking at the HLA, flow cytometry, and a PCR genotype PCRs of the looking for the donor NK cells in the tissues, and that's the next step in this endeavor. The CD16 expression is higher in a donor versus recipient NK cells. So red are donor NK cells. So these cells are actually having better CD16 expression, hoping they have a better ADCC. So that's positive. And also they do grow better than the patient's own recipient NK cells, as you see here with the expression of the Ki67 donor versus recipient. What we are looking for is, of course, the regulatory T cells. With this particular trial, we use a low-dose IL-2 and only for 7 days, so we have made some modifications there. And for most patients, we actually did not see the expansion of the regular T cells. And what is also very interesting to me and intriguing, then these 3 patients who did not have expansion of the regulatory cells are those patients who actually achieved a complete response with this therapy. There are 2 patients, and both of them had myeloma, so this is early to say what really it means. We have to really keep looking for these associations, but those are the patients who actually have a Treg expansion. And we did not find any toxicity, so I'm just going to show you a couple of examples of the clinical response. This is the positron emission tomography. It's a PET scan, which is using glucose attached to the fluorescent fluoride, and this is picked up by the active tumor, and usually it looks like a very hot spot when there's a high proliferation of the tumor. So this patient has an inguinal lymph node, which is really as big as a baseball, and this was most of his disease. And after 1 month post-treatment, you see the necrosis in the center of this tumor and significant shrinkage, and 3 months post-therapy. Cell infusion, there is more or less the scar tissue. We biopsy this tumor, there's only a necrosis, no viable tumor. This patient did not have any other therapy after NK cell infusion. He was just monitored. He was offered a stem cell transplantation, which he declined. The patient eventually relapsed about 5 months after this therapy and is undergoing some radiation. But so this is just interesting that the bulk of the disease probably doesn't matter. This therapy can affect even quite large tumors. The next patient is again with a follicular lymphoma at dose level 2. This is the tumor in the retroperitoneal lymph nodes, and this patient also was able to get in a complete remission. And this patient did eventually go for allogeneic transplantation. So we look at this therapy as probably a bridge towards more definite therapy, but it remains to be seen. So in conclusion, this story continues. We know that this is feasible and effective for some patients. There are no dose-limiting toxicities, and there are very, preliminary efficacy is observed, but much more needs to be learned. So with this, I'd like to introduce you to a new concept, and this is with a collaboration with another pharmaceutical company, and this is quite interesting therapeutic technology. It's based on a chimeric antigen receptor modification, and you all probably heard about the ability to reprogram the T cells by introducing the new DNA, which expresses on the new antigen receptor on the surface of the T cells, and then redirect the T cell towards the tumor. This concept, which I'm going to talk to you about, is called ACTOR, also antibody-coupled T cell. And this is using the receptor on the surface of the T cells, which is CD16, which is really the receptor which binds the Fc fragment of the antibody. So it contains a high affinity CD16. The DNA contains this CD16. It contains the co-stimulatory domain CD28 and then a CD3 zeta to link with the TCA signaling. And this piece of DNA is in a retrovirus which is inserted into the T cells through the genetic engineering, and the T cells are then grown ex vivo. So this is the patient's own T cells are redirected, and the specificity of a tumor is defined by the antibody you combine with this cell. So the beauty of this is that you can really use different antibodies for different tumors, and also can use those in sequence combination, and this is one of those how you can prevent the resistance. As we learn, one of the reasons why the CAR T are not effective in all patients is development of the resistant clones, which downregulate the antigen against which the CAR T cells are developed. So, for example, these ACTOR T cells are here combined with the different antibodies for hematologic malignancies and solid targets. These are the different cell lines, and these are in combination with the different antibodies. For example, you have a breast cancer cell line, use trastuzumab or HER2 antibody, and in combination with ACTOR, you can induce the IL-2 secretion from these ACTOR cells. If you use a target which is lymphoma and you use rituximab, exactly the same cell can induce the killing of and activation of the T cells. So that's why this is very much more flexible and potentially clinically very useful. So this is the process of developing the product. Each CAR T product is custom made and patient specific. The patient undergoes apheresis. These are from the patient's own T cells. Patients undergoes apheresis. The T cells are separated, and they are co-stimulated on artificial APCs and cytokines. They are co-incubated with the retrovirus, which contains the DNA of our interest, and then transduced, again grown for about 14 days, and eventually come in a 10 milliliter product to our patients. So it's a very complex process with a very unimpressive result. But what happens in the patient is quite amazing. So this is some preclinical data. The ACTOR cells, which are the CAR T with the CD16, are incubated with the rituximab, and they are specific for the Raji targets. However, they would not kill, any other targets, and they would not kill Raji if they are combined with the trastuzumab. This is the specificity. Here is the rituximab concentrations versus the IL-2 production by the ACTOR cells. You have to reach certain level at which the activity is then ensured. And this is just looking at the summary data from the preclinical animal models, looking at the mean photons and the fluorescence of the luciferase model. And these yellow are the lack of tumor in the patients who are treated with the ACTOR cells with the rituximab, as opposed to those mice which were treated with the rituximab only or with the ACTOR cells without rituximab. So this preclinical data led to development of the clinical trial. This clinical trial is now underway. We have presented our first data in a ACCR meeting in New York, which was great fun because I got to meet James Allison. He was there when he was presented with the Nobel Prize. So it was a fun meeting to be, but we have presented this data there. The process of manufacturing is very successful, and here are the expression of the CD16 on T cells. So this is a true chimera, right? T cells, they have all the TCRs or the receptors, plus they now artificially express CD16, which is really a chimeric manipulation. The biomarker of the efficacy is expansion, and this is the expansion in those six patients. The patients with the yellow are those who responded. With the black are those patients who did not respond. And as you see, this is giving the patients are giving lymphoproliferative chemotherapy and an infusion of the CAR T here, and then the actor cells expand rapidly within the first week. They of course have to be infused with rituximab, and in order for these cells to remain alive, we are giving rituximab every 3 weeks, which really stimulates their proliferation. And the actor cells appears to be persistent for a long period of time. We also look at some cytokines, and particularly IL-6, which is one of the cytokines which usually is very high in CAR-19 T cells, and is also clearly correlates with the development of the cytokine release syndrome and a clinical symptomatology of how sick the patients can get. With this product, we do not see IL-16 in those patients who are so far treated, and also the interferon gamma is really not very high. And that indeed also is reflecting in the fact that we did not see any CRS or neurotoxicity in any of these patients yet. It's certainly very early data, and this is not published only in abstract form, but this is not known and very intriguing to us that despite the fact that there is no IL-16 and cytokine expansion, the cells are actually functional and are proliferating. And the functionality is best seen by the clinical responses. This is one of the patients who, again, is the tumor is depicted here in the inguinal and iliac lymph nodes, which are hot on this scan, and after therapy, 40 days after therapy, these are all melted away as a result of the actual therapy with rituximab. So this is a patient with a grade 3 follicular lymphoma, and 2 other patients responded. We have seen some clinical response, although the duration and is really still too early to say. So this is bringing me to my last technology I'd like to review with you, and how we are using this in lymphoma malignancies. You certainly know Jeff Miller and Dan Valera. They are 2 great collaborators and friends who have been working on developing bi- or tri-specific natural killer cell engagers. The single fragment, single Fv fragment from the antibody here. This is an agonist anti-16 antibody, and this is the antibody against the tumor of your choice. In this case, it's a CD33 for AML. Linking these 2 antibodies in this form forms the small molecule which is able to link the natural killer cells with the tumor directly and cause the immunological synapse and allows the NK cells activation and redirected lysis of the tumor cell. And I've been working with Dan and Jeff to develop this for the lymphoma malignancy. So instead of the myeloid antigen, we are using anti-CD19 and anti-CD22, and even a tri-specific expressing both of these on the in the same molecule, which is a little bit more difficult than we thought, but still trying to make it work. So these bikes are then being improved by the insertion of the IL-15 and replacing the inroad linker with the fragment of IL-15 cytokine, which is functional and which is then causing the NK cell proliferation. So in our preclinical work done in a collaboration with Martin Felice, from Miller's lab, we show that this 16, 15, 19 trike is indeed causing the very nice, improving the killing of the NK cells and resulting in interferon gamma production after the NK cells are incubated with the 16, 15, 19, which is even better than rituximab. And more importantly, these NK cells not only kill, but also they proliferate. And this is shown in here. This is a proliferation and a viability, and this is the CSFE assay, which with a trike, we see a very nice proliferation, really confirming the functionality of the IL-15 piece. That IL-15 is functional and it's inducing the growth and proliferation of the natural killer cells. In terms of the functional assays, we look at tumor killing in this IncuCyte imaging assay. We co-incubated the Raji with a trike versus rituximab in an assay and look at a picture every 15 minutes over 44 hours. And this is the percentage of the alive cells in these cultures. As you see, the best killing is seen with a 16, 15, 19. And this is a combination with a no treatment, and the rituximab, I feel, is this purple. So it's actually is significantly better than rituximab. With this preclinical work, we are now doing some animal experiments, moving to a camelot CD 16, 15, 19, which really is a result of work by Martin, where the camelot, so this is the camel, the antibodies from the camel, can recall now the species that is much more functional than a human FC fragment, and replacing the human with a camel FC, we seems to have a better efficacy of the bike and trike. So with this, this is just slightly another experiment which is showing a stronger NK cell mediated killing, and this is for the primary tumors, CLLs, from the patients where the CLL, their blood tumors, indeed showing that the CLL killing is actually quite exceptional, and these are the CLL cells, lymphocytes. This is prior to culture and after culture. 66.9 cells are not alive. So with this, there's a lot we learned, but there's so much more we have to learn. And we have to bring many of these ideas back to the bench and back to a lab and trying to understand and learn from every patient what is this, why this patient responded and why this other one, the others not. And there are three different categories which we can really try to improve. We are working on improving the NK cell products. As I explained to you, the NAM NK is one of the products. We are also working very hard on developing inducible pluripotent stem cell derived NK cells, which would be off-the-shelf product. As you see, this is very cumbersome to have a specific product for each patient made. It's also very expensive. So off-the-shelf product would be a holy grail of cell therapy, and that's many companies, including our center, is working on this. Then how do we manipulate them in ex vivo to try to increase the cell numbers, but not to allow the patient exhaustion? We look at the host, and what is about the host immune system, how we can deplete Tregs, how we can deplete myelosuppressor cells. Should we combine them with the agonist? And then maybe there is a way to combine them with the checkpoint inhibitors or other antibodies which would inhibit those immunosuppressive elements. And then how we can better visualize the targets. Some targets are for lymphoma, they are better. For malignancies, they are more difficult. So can we maybe use the radiation to really increase expression of the neoantigens? That's one of the ways which has been combined with the CAR T cells, which is one of the ways which you potentially can make the tumors more immunogenic. So there are different ways how we can improve the immunotherapies. I think we made a great stride. What I showed you was a result of a last 8 works of my clinical and translational work here at the U. And I'm very thankful to many of my collaborators, mostly to Jeff Miller, who really I work very closely with, and he's my mentor. I was in his lab for a few years. Martin Felice, who is working, he's a PhD working on all this preclinical work. Sarah Cooley, and Dan Weinstrom, who is a clinician and who has been our chair for many years, Bruce Blazer, then of course, the patients and families, other patients who are brave enough and have the courage to go through the clinical trials of this complexity. The clinical trial office, I work very closely with the research nurses and CRAs and a translational therapy lab, which more of these laboratory specimens are analyzed. MCT and GMP facility, phoresis center, and then my collaborators from the Gamida and from UNUM, and then all my sponsors. And thank you for attention. I'll be able to take questions.
We can take questions from our remote sites, but let's start in this room if anybody would like to start. While I'm walking over, I'd like to ask about the map and why we are so susceptible to this.
Yeah, that's been, I think, a target of many epidemiologists here at the University of Minnesota and Mayo Clinic. At this time, we really cannot convincingly relate any causality or any specificity to increased incidence. There were work on potential, because if you know, there's kind of a belt in the northern. One of the possibilities was the vitamin D deficiency. So there was a big work done on that, and there is some association with the lack of vitamin D with some increased case of susceptibility, but it's not convincing. Whether it is to do with our industries, iron mining or other industries which are in Minnesota, there's a lot being done, but I don't have really a convincing answer for you.
Zohar?
I have a very naive question, and I apologize, but do we know what the T cells are doing, what the T-regs are doing to the NK cells?
Yeah, we've tested it in vivo. Yeah. So we co-inhibited those T cells from the patients with the NK cells, and they're inhibiting their proliferation. The mechanism is still, there are several different potential mechanisms. One is through the cytokine, the TGF beta and IL-10. The other possibility is through a direct inhibition. We've done several experiments. I cannot now, but they are functional. This is not only the number, but these Tregs are clearly functional. Most of it is through the cytokine release. And we have shown actually even serum of these patients have higher levels of IL-10 and TGF beta.
I have another question, if I may. So we always think of the NK cells as a bridge to transplant, and transplant is curative, but NK cells aren't.
Yeah.
Why aren't they?
Well,
do you know?
So this is a great question. I think the field is really evolving, and how is this understood, and it's with increasing experience. We thought, and the CAR-T, when they were developed, thought was that they are living drug, and they indeed have the persistence is necessary for ongoing clinical benefit. And for some diseases, it can be it's probably a case, such as pediatric ALL, where it was the first disease where the CAR-T was approved for. Their persistence is in a very, very low levels. It can be less than 0.5%. These transgene copy numbers are very low, but they are detectable, and they would be possibly expanded if there is an increasing of the antigen, such as relapse of the ALL. But in some of the more recent results, especially from the NCI by the co-inventor and also from the PEN, there is more understanding in the lymphoma that these CAR-T cells may not persist. And still, the patients for right now, it's about 2 to 3 years follow-up in some of the studies. I was involved in what's called the Juliet study with a CAR-T for the lymphoma. We have a median follow-up of 3 years, 2.5 years, and 40% of patients remain in a complete remission, and many of them, 2/3, do not have CAR-T detectable anymore. So it remains to be seen. But one of the possible explanations is that instead of the persistent, what you really need is that very high peak, that the peak of the CAR-T cells, and having the initial fight with enough toxicity and release is enough for the tumor elimination. It may be true for some tumors, as we know, some are even responsive to chemotherapies, and one cycle may be, or several cycles may be enough to eliminate. But for some cancers, the late relapses are very much a possibility, so I think it remains to be seen. But for NK cells, I think our first goal is to try to expand them and to really allow that first line. And whether persistence is necessary, it's still unknown. Yes, Bruce?
Give me a minute so that the people in the other locations can hear you.
I had a question about the CD16 CAR, and since that's going into T cell populations other than cytotoxic T cells, is it known whether it has any function in maybe like the CD4 T cells, which again aren't cytotoxic?
It is going to both CD4 and CD8. It is. I mean, this insertion is random. These are CD3 cells, so there is a combination of 4 and 8. There is for this particular CAR, I don't think we know whether CD4 expressing actor cells are functional or whether they have the same function. From the regular CAR19 field, we know that the CAR19, which are CD4 positive, are as functional as CD8s. There is really a quite substantial body of evidence that the CD4 are very functional. In fact, the group from the Seattle, Fred Hutch, is using a very well-controlled ratio of CD8 and CD4 to achieve a perfect mixture of these cells. In terms of the CD16 expressing actor cells, it's a good question. I don't think we really know what the CD4 T cells are working. Well, what do you expect?
I was hoping you could answer that.
Do you retreat at all if you see a CR, and is there any limitation to how many times you could retreat? And then my other question is, I'm just curious from a clinical trial standpoint, how much money it costs to put a patient through some of these therapies?
Yeah, so we just, a very good question. We have actually amended the protocol to allow retreatment, and only one time. We want 3 months a gap in between the treatments, but for the patients who have benefited, we would allow them retreat. We don't want to retreat too many times just because it's costly, and this is not a therapy which meant to be repeated. It is really very expensive. The product for the NK cell, for example, for the non-NK cell, cost about $30,000. For the actual cells, it's probably even more expensive, but as you know, the commercial CAR T cells are very expensive. They cost $350,000 per patient, just the product itself, $350,000. Okay.
Can I ask one more little question? Can you remind me, too, what the correlation with CMV positivity is and from the donors?
In my work, we are not looking at a CMV positivity, or we are not really looking at a serostatus of CMV, but we've demonstrated that the patients who are CMV positive, and particularly if they have a CMV activation, especially post-transplant, those patients have enhanced proliferation of the NK cells which have an adaptive phenotype expressing NKG2C and CD56. This is a type of NK cells which we described, and they are particularly interesting because they have a higher function, better killing, and better persistence. So with that knowledge from the transplant world, we brought that to this platform, and now expanding the adaptive NK cells from the seropositive donors as a way to enhance their function in patients. Yeah, good question.
Very good. Any other questions or comments? All right, let's thank Dr. Bachanova.
Perfect timing. Thank you.